Thank you very much for the introduction. What I'm going to do, um, so it looks like I can't share my screen. Is there a way to be able to do the screen share?
Please,
Thank you. Uh,
maybe at the bottom, you may click.
no, it says host disabled participant screen sharing. Let's see. Yeah, I know that. You have slides that can be used. The slides I have here are a little bit updated. So I don't know if there's a way for me to share the screen. I see in the chat they're hopefully getting something ready to go.
So,
Hmm. Well, we can go, I guess, with the slides.
yeah, so it does not work for the slide share.
It doesn't. It says the host disabled participant screen sharing.
Really?
Um, well, we can go with the slides that I sent. They should be fine. It's just a little bit difficult here. Oh, he says stand by, so hopefully that will change in a minute then. Okay, good. Looks like we're going now. Okay, let's see if that works. Good. Can you see that screen okay?
Yes, yes, very good.
Okay, great. Good. Thank you. All right. Well, no, thank you for the opportunity to make some comments this evening to the conference. In talking about therapies for COVID-19, it's quite a challenge given the fact that the literature in this area is extremely vast. Early on in the outbreak, going back to the early part of 2020, a large number of studies rapidly emerged, ended up in a variety of public domains. A large number of studies were initiated. And again, while studies had begun in China and some useful information came from that, as COVID-19 hit the more global scene, it really became a large number of studies with little uniformity in patient eligibility, primary and secondary endpoints, looking at things such as time to recovery, movement on an ordinal scale, organ support-free days, mortality. The sample sizes from these studies range from single digits to thousands of patients. Study designs ranged from observational studies to open label trials with historical controls, to randomized open label studies, to randomized placebo-controlled trials. We have not had, although I think we're getting closer to having, clearly validated laboratory markers for predicting clinical outcomes. Complicating things even further is the fact that the spectrum of illness and underlying pathophysiologic processes associated with SARS-CoV-2 infection are not well understood. And there certainly has been no lack of experts with very strong opinions about what one should or should not do in the treatment of these patients. This slide just tries to depict some of the different clinical manifestations one may see in the setting of SARS-CoV-2 infection, ranging from no symptoms to outpatients with mild symptoms, to inpatients requiring no oxygen, low flow oxygen, high flow oxygen, or even mechanical ventilation. The therapies that are being studied in COVID-19 range from antiviral strategies, which appear to have their best efficacy early in the course of illness. I'd include the monoclonal antibodies in this group, to the immunomodulatory strategies that appear to be most effective later in the course of disease. And then throughout the course of illness, there are concerns about the role of coagulation in tissue damage, and anticoagulation studies are being looked at basically every stage of SARS-CoV-2 infection. Fortunately, there were some large platform trials from which a lot of the data upon which treatment guidelines are based were able to be started. These included in the US, the ACT-1 trial, which was a series of randomized placebo-controlled trials, the recovery trial in the UK, and I know you've heard from Professor Horby on this, the Solidarity trial launched by WHO, and then the active series of studies, a public-private partnership by US government agencies with a variety of pharmaceutical industry partners. Just on here is the most recent graph I had of the global outbreak, and as of November 9th, with just over 252 million cases and 5 million deaths. So it remains a very serious problem globally. Just a few words about the virus before talking about treatments. SARS-CoV-2 is a beta coronavirus. It's the same subgenus as SARS-CoV-1 and closely related to some bat coronaviruses. It is an RNA virus. It has a relatively large genome with 4 structural proteins. The spike or surface protein of the virus is the target for the current monoclonal antibody therapies. This is where the virus binds to the host cell using the ACE2 molecule. You can see a cartoon of the virus on the right, a transmission electron micrograph in the insert, and then the X-ray crystallographic appearance of the protein in the far right. In terms of the life cycle, when the virus enters the cell with some uncoding, exposing the viral RNA, that is then translated into a polypeptide, which is cleaved by the viral protease, leading to the different components of the viral replication cycle, allowing for the RNA-dependent RNA polymerase activity to make genomic copies. Again, the antiviral strategies today look at polymerase, protease, and then the monoclonal antibodies interfere with inhibition. So again, with time, we're developing more and more tools that allow us to attack this virus directly. A couple of laboratory tests I want to mention because I think going forward, some of these measurements of viral proteins may become more and more important. This is a technology developed by a company, Quanterix. It's a very sensitive way for detecting the nucleoprotein, the nucleocapsid of SARS-CoV-2. It's a magnetic bead antibody capture assay. It deals with this disc that captures a single bead. So it becomes a very sensitive way to measuring amounts of antigen, either in nasopharyngeal swab fluids or actually even in plasma. And I'll show you a bit of the plasma data in a moment. So this is one way to measure plasma antigen of SARS-CoV-2. This is looking at the sensitivity of the test. In serum on the right, looking at spiked samples, being able to detect somewhere in the range of 0.01 TCID50s, tissue culture infectious dose 50%, so a very low number of virions. And indeed, as shown on the right, even in patients who are PCR negative, this is from a small cohort from some of the initial data on this platform, even in patients who are PCR negative, you at time will see positive antigen for SARS-CoV-2 nucleoprotein. A whole array of antibody assays are available in the United States. There are many assays that are being used under emergency use authorization. The authorization is for detecting prior infection with SARS-CoV-2. They aren't really authorized for use in monitoring or segregating patients with regard to response to therapy, but they do seem to have some potential in that area. Two main types of antibodies, those that antibody assays, those that measure antibodies to nucleocapsid, and the one that we've been using most frequently measures IgM, IgA, and IgG. We also are using the same technology that we use to measure antigen, to measure anti-spike antibodies. This is just looking at IgG antibodies to spike. And then the third assay we use is a surrogate for viral neutralization. It's a competitive inhibition assay looking at competing with the binding of ACE2 to the spike protein. If antibodies are present to the spike protein, they inhibit the binding, shown here in the orange. So this is a pretty good surrogate, and in terms of comparative assays, it's a pretty good surrogate for pseudovirus neutralization or virus neutralization, and it's a much easier assay than amenable to higher throughput. We've been using these assays to try to look at predictors of recovery. Here I'm showing some data from a study referred to as TICO or Active3. I'll talk about a little bit more in a moment. It's a randomized placebo-controlled trial of monoclonal antibodies in patients who are hospitalized with COVID-19. This particular sample is a group of about 320 so patients looking at 3 different biomarkers: anti-receptor binding domain antibodies, so that would be the gene script pseudo-neutralization assay, anti-nucleocapsid antibody, and then plasma antigen level. In each instance, we've bifurcated the results basically by half. So antibody positive or negative for the 2 antibody assays, and then plasma antigen levels less than 1,200 picograms per ml or greater than or equal to 1,200 picograms per ml. And I think you can see, the risk of having recovery is increased in those patients who have higher antibody levels at baseline or lower plasma antigen levels at baseline. So these appear to be pretty good biomarkers then for predicting outcomes here in the placebo group. And as I'll show in a moment, that helps bifurcate, I think, patients who may or may not respond to monoclonal antibodies. So moving now to talk about the specific therapies, broken it into antivirals, immunomodulators, and anticoagulants. Among the antivirals, there are the monoclonal antibodies to the SARS-CoV-2 spike protein, a series of RNA polymerase inhibitors. I'll talk a little bit about Remdesivir and Molnupiravir, and then protease inhibitors. I'll talk a little bit about the Pfizer drug that was recently reported in the media last week. Regarding the monoclonal antibodies, these appear from a variety of studies looking at several different monoclonals to be really quite effective when used early in the course of illness. They have been shown to be able to decrease the 5 to 7% rate of progression of ambulatory patients at risk for severe disease progression, to decrease that rate from 5 to 7% to 2 to 6%, depending upon what study you're looking at. So that is a relative reduction of about 70 to 85% in the rates of hospitalization or death. So this really is, I think, something of considerable value for patients at high risk of disease progression. They've also been shown to be of benefit in a post-exposure setting. Overall, they've not been of benefit in hospitalized patients, but in data I'm guessing you may have seen from Professor Horby in recovery, and in some data I'll show you from TICO, that there may be a subset of hospitalized patients where in fact they could be of help. In the US, we have 3 different monoclonal antibodies available for use under emergency use authorization. Bamlanivimab combined with etesevimab intravenously. This combination is less effective against the beta and gamma variants of SARS-CoV-2, and at the point in time in the US when those variants were more prevalent, we actually were not using this combination. Delta is quite sensitive to this combination, so now it is a combination used basically and should be effective anywhere where Delta is the major variant circulating. The next combination is the combination from Regeneron, casirivimab, and imdevimab. This can be given intravenously or subcutaneously. It's a combination of a human antibody and an antibody from a humanized mouse. It's authorized both for treatment and post-exposure in the US. And then the final antibody from Vir GSK, sotrovimab, which is given intravenously. It's a human monoclonal, interestingly, that was developed to SARS-CoV-1 and has cross-reactivity with SARS-CoV-2. The study that we've been most involved in at the NIH looking at monoclonal antibodies has been a platform trial as part of the active portfolio of studies, ACTIV-3, a multi-arm, multi-stage trial, where instead of doing separate randomized controlled trials of different agents, they're all combined into one study. Patients are randomized to a control, which thus far has been a placebo control, and one or more different interventions. There's an initial evaluation after about 300 individuals have been enrolled, and unless there's some hint of efficacy, that agent is stopped at that point in time. If there is some hint of efficacy, it goes into a more classic phase 3 type evaluation. This shows the data looking at bamlanivimab as a single agent in hospitalized patients with an endpoint of time to sustained recovery or time to hospital discharge. And I think you can see pretty easily from these two graphs that neither of these endpoints showed any difference overall for this population of hospitalized patients, whether they received bamlanivimab or a placebo. Interestingly, though, if you looked at some of the subgroups from this study, either negative or positive for neutralizing antibody, high or low levels of antigen, or a combination of antigen and antibody, I think you can see differences in the results with p-values for interaction pretty close to significance, overall suggesting that patients with lower levels of antibody at study entry and higher levels of antigen at study entry may be those most likely to see benefit. And additional work continues on using this paradigm and now studying additional antibodies. The most recent one that we're looking at is from a combination from AstraZeneca. Again, those data I think are very similar to the data I'm guessing you may have heard from Professor Horby from the recovery trial, which had as one of its arms a randomization to casirivimab or imdevimab in overall, but the subset analysis in antibody-negative patients indicated that indeed, while there wasn't a clear difference in mortality overall, there was a hint. If you separated that by seronegative versus seronegative, you did see a pretty striking result. So I do think we'll see, and I know WHO has as part of the treatment guidelines, the use of this monoclonal for hospitalized patients who are antibody negative. Move now from talking about antibodies to talk about some of the small molecule antivirals, starting with the small molecule inhibitor Remdesivir. This drug is licensed in the US for treatment of COVID-19. It was shown to decrease time to recovery in a randomized placebo-controlled trial known as ACT-1. It had no effect on in-hospital 28-day mortality seen in an open-label randomized trial, Solidarity by WHO, and was found to have decreased mortality and hospitalizations in an outpatient study. So this is a little bit more controversial, I would say, than the data I presented on monoclonals, where I think most people have interpreted their efficacy in a very similar fashion. For Remdesivir, it does appear that based on study design, perhaps based on what background therapies were, some studies have shown benefit, others have not. As I mentioned, it is something that is licensed in the US and is part of our treatment guidelines. These data here are from the first study of Remdesivir in the US. The very first study on Remdesivir was a placebo-controlled trial in China that unfortunately was not able to reach its desired sample size. This study enrolled somewhere around 900 patients. It randomized them to placebo or Remdesivir. The outcome was time to recovery. Recovery was defined as reaching ordinal scale 1, 2, or 3, which was not hospitalized at full activity, not hospitalized, limited activity, or hospitalized for non-medical reasons after having been admitted, either requiring oxygen, not requiring oxygen, high flow oxygen, or mechanical ventilation with ordinal scale 8 being death. So again, you could see a pretty nice separation of the two curves regarding time to recovery. This is in contrast to the data from the Solidarity trial that looked at 28-day mortality and found that, in fact, they saw no difference between remdesivir or control. Similar data were generated by that trial for hydroxychloroquine, lopinavir, ritonavir, and interferon beta with or without lopinavir, ritonavir. Again, if you looked at the Solidarity trial based on severity of the patients at study entry, it appeared that there was a trend towards harm for those patients who are mechanically ventilated, and perhaps a trend towards some benefit in not ventilated patients. A lot of study is going on trying to parse out where remdesivir may indeed have its greatest benefit. Looking here back to the Act 1 trial on mortality, if you look overall on mortality, you see a hazard ratio of 0.73 with a 95% confidence intervals from 0.52 to 1.03. So no statistically significant impact on death overall. However, if you look at some of the subgroups that were not adequately powered to show clear results, you do see that the best hazard ratios for preventing death are seen in the patients who were either not receiving oxygen or on low flow oxygen, whereas there, again, as in the case of Solidarity, a hint that there may actually be some toxicity of the drug for patients on mechanical ventilation. So again, it appears that early in the course of illness, even in hospitalized patients, remdesivir may have some benefit. A recent study that came out in presentations at meetings and in a press release from the company Gilead, they showed in outpatients are randomized to receive remdesivir or placebo with the primary endpoint of hospitalization or all-cause death by day 28. That treatment with remdesivir reduced hospitalization or all-cause death by day 28 with a hazard ratio of 0.13, a statistically significant result. Again, similar to the data from monoclonal antibodies, at about an 87% reduction in hospitalization or death with treatment. Just make a couple of words about molnupiravir. This is an RNA polymerase inhibitor, relatively broad spectrum activity. In a press release from the company Merck, working with the company Ridgeback, 762 patients randomized 1-to-1 to receive molnupiravir or placebo. Primary endpoint, again, hospitalization or death. Treatment with molnupiravir significantly reduced hospitalization or death by day 29 by about 50%, 7.3% in the treatment group versus 14% in the control group. And there were 0 deaths in the molnupiravir group compared to 8 deaths in the placebo group. This drug is currently under review by the U.S. Food and Drug Administration for emergency use authorization. I think they're planning an advisory committee meeting on November the 30th. The other drug I want to mention are the protease inhibitors being developed by Pfizer. They have 2. This one, 7304814, is an intravenous drug. The one that was recently with the press release, 7321332, is an oral drug. This oral drug, Paxlovid, was the topic of a recent Pfizer press release. This drug also is being reviewed by the FDA for emergency use authorization in the US, and I think will be reviewed at that same November 30th FDA advisory committee. In this study, 774 patients were randomized 1-to-1 to receive Paxlovid or placebo. The primary endpoint was hospitalization or death. Treatment with Paxlovid within 3 days of symptom onset significantly reduced COVID-19 hospitalizations or death, 0.8% versus 7%, a highly significant result. Again, about that same, 80% or so reduction in hospitalization or death, no deaths in the Paxlovid group compared to 7 deaths in the placebo group. This is just comparing the data from these 2 oral antiviral agents that have been studied in ambulatory patients. So molnupiravir is an RNA polymerase inhibitor. Paxlovid is a protease inhibitor. The 2 studies both enrolled a bit over 700 patients. 50% reduction in hospitalization or death with molnupiravir versus 89% with Paxlovid. I just bring to your attention, though, that the patient populations are a bit different for these 2 studies, as presented in the press releases. You can see that the 14% of the control arm in the molnupiravir study reached an endpoint versus 7% in the placebo arm in the Paxlovid, suggesting that the molnupiravir study enrolled a slightly sicker group of patients. Again, I think we're waiting to see more data from the studies to know whether or not that's actually the case. But I think it's really good news that if we have 2 oral drugs that appear to have relatively good toxicity profiles that also look to be quite effective in preventing hospitalizations or death. Just to say a few words towards the end here on the immunomodulators and anticoagulants. These are the data I'm guessing you saw from Professor Horby on recovery, showing a very marked effect on dexamethasone compared to usual care for patients who were admitted to the hospital. Of particular note, though, is that if you looked at the patients who were hospitalized and not receiving oxygen, there was a hint, in fact, that the corticosteroid dexamethasone might actually be of harm. So again, this is something recommended for hospitalized patients not receiving oxygen. It is not recommended for use in ambulatory patients. In terms of other immunomodulators have been studied, prominent among them, JAK kinase inhibitor baricitinib, IL-6 blockade with tocilizumab or sarilumab. All 3 of those drugs actually are being used at times in combination with steroids in patients with advanced disease where inflammation is felt to be playing a major role in pathogenesis. Anticoagulants are also studied as therapeutics in patients with COVID-19. At a minimum, prophylactic dose heparin is recommended in all hospitalized patients. A series of studies have suggested that full dose anticoagulation in severely ill patients is not of greater benefit than prophylactic dose anticoagulation. So full dose anticoagulation is not recommended in critically ill patients other than if there's another indication, such as deep venous thrombosis or pulmonary embolus. For non-critically ill hospitalized patients, some data have been published suggesting benefit overall to full dose anticoagulation compared to prophylactic dose anticoagulation. If one looks at those studies, the eligibility criteria and the exclusions are quite numerous. And I think a lot of the guidelines groups are sorting through the data from those studies to see which patients without other indications for full dose anticoagulation who are not critically ill might in fact benefit from full dose anticoagulation. In addition to heparin, other forms of anticoagulation, some of the oral agents and others are currently under study. Among some of the things that have been studied and not under consideration would be convalescent plasma and hydroxychloroquine. Among the therapies for which the jury is still a bit out, ivermectin, fluvoxamine, montelukast, the protease inhibitor from Shionogi, oral versions of remdesivir, and then again, there are multiple other antibodies and immunomodulators currently in clinical trials. It is challenging to sort out all the data that are published on treatment regarding treatment for COVID-19. In the US, we've put together a treatment guidelines panel under the umbrella of the NIH to try to provide some degree of guidance. These are obviously not mandatory dictums. These are really guidelines to try to help clinicians sort through all the information that's out there. This was initially requested in March 2020. We got the first release out sometime in April. Since then, it has had enormous numbers of hits. When I made the slide, it was somewhere around 15 million. The latest number I had was just over 17 million page views. We split it into 2 main categories, recommendations for outpatients, where the key recommendation is using monoclonal antibodies in individuals at risk and not using corticosteroids. I think that's the big takeaway for outpatients. In terms of hospitalized patients, again, the use of dexamethasone or other corticosteroids for patients who are hospitalized and require supplemental oxygen, recommend against the use of steroids in patients not requiring supplemental oxygen. For those patients who are requiring supplemental oxygen, we also recommend consideration for remdesivir in their earlier stages of illness and the addition of baricitinib and or tocilizumab or one of the other IL-6 receptor inhibitors like sarilumab in patients who are doing poorly, particularly patients who show a rapid decline in their clinical course and or who have inflammatory markers. So with that, I'll conclude, and I thank you again for having the opportunity to make a few comments this evening. And I'll
Yes,
stop
thank you very much for your wonderful presentation. I'm sorry we are behind of the schedule, but let me see if there are any questions from the audience that luckily we do not have yet. So my final question is that now we have an oral COVID-19 drug from Merck and Pfizer, and what do you expect the future if they are available, or still do we need this kind of strict isolation or uh, or something like that, or can we treat this COVID-19 just like flu or something like that? So that's my question. Yeah.
It's a good question. I mean, with any of these therapies, as well as with the vaccines, we appear that we may be having an impact on the course of illness, but we may not be having much of an impact on the transmission of infection. So even though I think these drugs will have an impact on outcomes, I think, mortality rates, I hope will drop, hospitalization rates, I hope will drop. And I do think they will as these oral drugs get out there, as they have with the antibodies. I don't know that it's going to do much to prevent transmission. So I think we're still going to have a bit of a challenge until the overall numbers globally go way down.